Centriole and Centrosome. Centriole are the structures which are assisting the formation of spindle fibers, and the other tubular structures present in the animal cell, because here the centrosome or the centriole structures which are helping the formation of spindle apparatus and formation of bipolar spindle, and the instead of microtubules as like in the plant cells. So, they are instead of microtubules in the plant cells, the centrioles are the structures which are concerned with the formation of spindle fibers and also the movement of the chromosome. So, this centrioles are the structures which are present inside a nearly spherical structure called as a centrosome, and centrioles are minute submicroscopic structures which are surrounded by amorphous substance in a centrosome. So, if you see the outer view of this one, centrosome structure contains a spherical shape, nearly spherical shape structure which contains two cylindrical organizations. Those cylindrical organizations contains 9 peripheral fibrils. And these peripheral fibrils are arranged like this, and these two centrioles are arranged in perpendicular to each other, and each of them contains 9 peripheral fibrils. And surrounding these fibrils, it is surrounded by amorphous substances. So, this type of organization can be seen in the centrosome, where we can see 2 centrioles, each centriole with 9 peripheral fibrils, and here also 9 peripheral fibrils which are arranged in perpendicular to each other. This type of organization can be seen inside the centrosome, and surrounding this one, this part is contain amorphous substances. And if we take the transverse section of the centriole shows nearly cartwheel like organization. So, centrioles does not have 9+2 arrangement, but why? Because the central fibrils are absent here, but instead of the central fibril, it is having a proteinaceous core like structure. The proteinaceous core like structure is called as hub. This part is called as hub, and hub is connected to the peripheral fibrils. Here instead of dienes, so we have seen the dienes in the, that is 2 microbodies in the peripheral fibril structure, but here we can see 3. So, we call it as a triplet like organization. So, these are A, B and C. So, from inside to outside they are A, B and C, and here A contains 13 complete 13 protofilaments are present, because it is a complete structure, whereas the walls of A and B are shared by them, so that does not contain the complete 13 protofilaments. So, this type of organization can be seen. ABC, so here we can see 9 peripheral fibrils, but each of them is a triplet, because they are having A, B and C microtubular structures. So, here from the hub region, this is a hub region, central hub is present. From the central fibrils, 9 radial spokes will be arising. And at the end they have a thickening like structure. These thickenings are called as X, which are connected to the which are connected to one more thickening called as a Y thickenings. Then these are the radial spokes. These are called as X thickenings and these are called as Y thickenings. And here from A to C we can see some connection like structures. These are called as CA linkers. From C to A the linking will be present. This is called as CA link or CA bridge we can call it as. And here this Y thickening is connected to CA link. And this X thickening is connected to A microfilament structure or A microtubule of the peripheral microfibril. This type of entire structure appears to be like a cartwheel like organization, so we call it as cartwheel structure of centriole. Here we can see the presence of a central hub which is made up of tubulin protein, and from there we can see the formation of 9 radial spokes, and these radial spokes shows a thickening like structure called as X thickening. And on the peripheral side we can see the peripheral microfibrils, and each peripheral microfibril is a triplet of peripheral microfibril. And the A is a complete one which contains 13 protofilaments, whereas B and C are they does not have complete protofilaments because the walls are partially shared by them. And here the X thickening is connected to Y thickening, and this Y thickening is connected to the CA link. CA link is the one which is connecting the C microtubule of one peripheral fibril to the A microtubule of the adjacent microfibril. So this Y thickening is connected to the CA link, and the X thickening is connected to the A microtubule of the filament. So this entire structure appears to be like a cartwheel like organization. And on the peripheral side we can see the maculae. On the outer side we can see the maculae, 9 maculae are present. So these are called as maculae or pericentriolar satellites. So these are the structures which are present outside the peripheral centriolar structures. These are acting as a nucleating structures during the astral formation by the centrioles, because the centrioles or centrosome structure does not have the nuclear organization, so that this maculae or pericentriolar satellites is acting as a nucleating structure while the formation of the astral spindles, and that is formation of bipolar spindle during the movement of chromosomes, or which is helping in the attachment of spindle fibers to the centromere region and the movement of the chromosomes. So this is a organization that we can see in the cartwheel like organization of the centrioles which are present inside the centrosome.